Hello, my name is Tariq Adar, and today I'll be discussing G protein coupled receptors, specifically the GS and GI, G stimulatory and G inhibitory G protein coupled receptors. So here we have the G protein coupled receptor, the subunits of interest, the alpha, beta, and gamma subunits. The alpha subunit is involved in one signaling process, and the beta gamma subunit is involved in a separate one. So at the end, they're both activating the effectors of the signaling pathway. So it begins when the ligand is going to bind to the G protein coupled receptor. An important example that you must know is glucagon. All right, glucagon binds to a G protein coupled receptor as opposed to insulin, which is going to use a receptor tyrosine kinase. All right, that's an important distinction that you need to be familiar with, and examiners can test it. All right, so glucagon is going to bind to its G protein coupled receptor, and the alpha subunit is going to dissociate. Why? Once that G protein gets bound, it's going to act like the guanine exchange factors that we talked about in the last lecture. So it's going to exchange, so this gets activated and it's going to swap the GDP for GTP. Okay, this gets activated and we're going to swap GDP for GTP. That is going to cause a conformational change so that the alpha subunit is going to dissociate. And so the G protein is going to come off the receptor and the beta gamma subunit is going to come off of the whole complex. All right. And of particular interest to us when we talk about G stimulatory and G inhibitory, the GS and GI proteins, the most important effector that we need to discuss is cyclic AMP. All right, cyclic AMP is the prime second messenger for GS and GI signaling. All right, so ATP is usually present in the cytosol of the cell. And adenyl cyclase is an enzyme. Adenylyl cyclase is an enzyme. All right, and it's going to convert ATP into cyclic AMP. So it's in the name, adenylyl cyclase. All right, and that can be broken back down into 5' AMP by the action of phosphodiesterase. Okay, so to repeat, you have ATP present in the cytosol of the cell. Normally, adenylyl cyclase will generate cyclic AMP from the ATP and then it will be degraded by phosphodiesterase to 5-AMP. Now, G-stimulatory proteins are going to increase the activity of adenyl cyclase. All right, G-stimulatory proteins are going to increase the activity of adenyl cyclase. G inhibitory proteins, so GI proteins, are going to increase the activity of phosphodiesterase. So in other words, GS is going to increase the activity of adenylyl cyclase so that you get more cyclic AMP. And cyclic AMP phosphodiesterase is going to break down cyclic AMP so that you have lower levels. All right? So a G-stimulatory protein stimulates changes in the cell by increasing the levels of cyclic AMP because cyclic AMP is going to act on protein kinase A, which we'll talk about in a second. All right, and then this is going to cause the downstream effects. Whereas phosphodiesterase is going to break down cyclic AMP, and that's why it's associated with the inhibitory G proteins. All right, so let's say you have a genetic defect in the GS protein, all right? Whatever enzyme or whatever hormone was supposed to be active on that G protein, you're going to have decreased function. So you'll have metabolic disturbances, brain issues, especially there's some forms of insufficiency for thyroid hormone and other hormones. The three that I want you to really know as that their responses are mediated by cyclic AMP are going to be the heart. Okay, so increased cyclic AMP in the heart is going to increase the heart rate and the force of contraction under the action of adrenaline. I want you to know that glucagon acts on the liver to break down glycogen, because glucagon wants to make more sugar available in the blood. So glucagon acts on the liver to promote glycogen breakdown, and vasopressin is going to act on the kidney to promote water resorption. Okay, vasopressin is released when the body detects not enough water in the blood, and it's going to cause water resorption. Those are three examples that you probably should be familiar with. okay? Because they are classic hormones. Adrenaline, glucagon, and vasopressin are very classically tested when it comes to their mechanism of action. All right, moving on. Here you see the effect of cyclic AMP. So you see there are these regulatory subunits that are bound to these regulatory regions of protein kinase A. So this is protein kinase A. All right, I'll write it here for you. This is protein kinase A. And it has these regulatory regions that are bound by a regulatory subunit. And cyclic AMP binds to the regulatory subunit to lead to active catalytic subunits because these come off, okay? So these were sequestering protein kinase A. And when they are bound by cyclic AMP, it frees the protein kinase A so that it can go on to serve its function. So go ahead and take a moment to read the question and then pause. So the question was, which of the following mutations would not cause an overall gain of function in the G protein coupled receptor pathway? So let's go one by one through these and see whether it's going to cause gain of function. In other words, at the end of the day, will there be increased protein kinase A? So when we say gain of function in the pathway, we're looking at the end point, which would be, is protein kinase A going up? If protein kinase A is going up, that means you have gain of function. All right. So the first option is going to be a gain of function mutation, so an activating mutation in the alpha subunit. All right, if the alpha subunit increases, adenylyl cyclase is going to increase in its activity. You're going to have more cyclic AMP, and because you have more cyclic AMP, you're going to have more protein kinase A activity. Why? Because cyclic AMP inhibits the regulatory subunits that prevent pKa. Now, what would happen if we had a loss of function mutation in the pKa regulatory domains? So if cyclic AMP is meant to inhibit the regulatory domains, if the regulatory domains didn't work, they had a deactivating mutation, then protein kinase A would always be free to have its effect, right? Because the regulatory subunits are supposed to bind protein kinase A. If this is pKa, these are the regulatory subunits, all right? This is another pKa. And then let's say the orange, this is cyclic AMP. Normally cyclic AMP bounds to the regulatory region, and when it's bound to the regulatory region, pKa can come off. But if the regulatory region doesn't work, protein kinase A becomes constitutively active. All right. A gain of function mutation in phosphodiesterase. Okay, so where does that fit into our balance? Phosphodiesterase is going to break down cyclic AMP. When phosphodiesterases are overexpressed or have gain of function, we're going to have decreased cyclic AMP, which is going to lead to decreased protein kinase A because you won't have binding of the regulatory region so that the protein kinase A can't come off of the regulatory region. So this is a gain, so this would cause loss of function. So this is loss of function in the GPCR pathway as an end result. So just to repeat, so far, this does cause a gain of function. This does cause a gain of function. This one caused a loss of function. A gain of function mutation in the phosphodiesterase makes it more active, which causes less cyclic AMP to be present because it's going to be converted from cyclic AMP to 5' AMP. So that gain of function of phosphodiesterase is going to decrease the amount of cyclic AMP, which is going to prevent the release of pKa from the regulatory regions. So a loss of function mutation in adenylyl cyclase would have a similar effect. So if adenylyl cyclase isn't working, you don't get the cyclic AMP. All right? You don't get the cyclic AMP if adenylyl cyclase isn't working. So it's the same thing as if you had gain of function of phosphodiesterase, which is an interesting observation. All right? Because what that says is enzyme A will cause the production of B, and then enzyme C causes the production of D. So let me actually make this enzyme A, and we'll call this X, which represents ATP. So you can get buildup of B through one of two ways. Either a problem with C can cause a buildup with B, or an excess of A can cause a buildup of B. Conversely, an excess of C can cause a lack of B, right? An excess of C that pushes this too much in this direction causes a lack of B, and a lack of A can also cause a lack of B. All right? Some G proteins use guanine instead of adenine, so a different purine nucleotide. So to treat pulmonary hypertension, we want to relax the blood vessels. Okay? We want to relax the blood vessels. What relaxes the blood vessels is going to be cyclic GMP. Okay? However, cyclic GMP gets broken down by phosphodiesterase enzymes. Okay? So cyclic GMP is supposed to cause relaxation of the vasculature. All right? It's supposed to cause relaxation of the vasculature. And phosphodiesterases break it down. So in patients with pulmonary hypertension, we want to increase the amount of cyclic GMP. We want to increase the amount of cyclic GMP. And what we can do is we can do that by blocking PDE. Remember that image I drew on the last slide? How I said if you have a process, so let's say we have substrate X, gets broken down to substrate Y and then substrate Z, you can increase Y by one of two ways. You can either activate A, okay? You can either activate A so that you get more breakdown of X into Y, or you can inhibit B so you get less breakdown of Y into Z and you can build up Y. All right? So we want to prevent cyclic GMP from being broken down to 5' GMP by phosphodiesterase. So what we do is we block phosphodiesterases. And that's the reasoning behind phosphodiesterase inhibitors. Because when we administer the phosphodiesterase inhibitor, and that can be sildenafil, Viagra, which might ring a bell as having a different indication, and we'll get to that in a second. But for example, tadalafil, sildenafil, etc., you can use those to treat pulmonary hypertension, because what they're going to do is that they're going to inhibit phosphodiesterase, and that's going to increase cyclic GMP. And because the cyclic GMP is increased, we're going to have relaxation of the vasculature, and that's going to ameliorate the pulmonary hypertension or make it less bad. Okay? Additionally, this can dilate the blood vessels in men for their erections, and that is why it can be used for that purpose. In fact, sildenafil, was commonly known as Viagra, was used initially being studied as an antihypertension medication, and they found that erections was the side effect. And the reason that was because it's a phosphodiesterase inhibitor that's going to increase cyclic GMP, and that increase in cyclic GMP will cause blood vessels to relax. All right. So the last clinical correlate we'll have is cholera and the pertussis toxin. So what the cholera toxin does is that it causes ADP ribosylation. Don't worry about that specifically right now. From intracellular NAD+, so you might remember that NAD+, it's important for glycolysis. It's just hanging around in the cytoplasm. What it'll do, it'll take a component of NAD, so don't worry about this phrase ADP ribosylation. What I want you to know, it's going to take a component from NAD and it's going to paste it onto the alpha subunit. And when it takes it from NAD and puts it on the alpha subunit, that subunit will no longer hydrolyze bound GTP. Okay? So when GTP is bound to the alpha subunit, it's constitutively active. Now, once the alpha subunit is constitutively active, we're going to have constitutive activation of adenylyl cyclase. So that's going to be switched on, and that's going to increase cyclic AMP. So we're going to get increased secretion of chloride from the GI tract. So the effect of increased cyclic AMP inside the GI cells, that it activates the secretion of chloride into the GI tract. So if this was the cell, right, and it has these pumps or these channels, the chloride is being pushed into here. And where ions go, water goes. So the ions get secreted into the lumen or opening of the GI tract, and the water will go with it, and what we'll get is rice water stool. and that is the mechanism of action of the cholera toxin. It ADP ribosylates the alpha subunit so that GTP is permanently active, and this will increase adenylyl cyclase, and this will cause increased secretion of chloride from the GI tract and lead to rice water stools. Pertussis also acts through ADP ribosylation, but of the inhibitory subunit, which is going to keep the G protein from regulating its targets. So a GI subunit is trying to keep things from acting, all right? So when you trap it in the GDP state, the GI, the inhibitory pathway, doesn't activate. Because the inhibitory pathway won't activate, right? So you get knockout of the inhibitory pathway, you're going to have increased secretions and decreased ciliary function. So the cilia that are in your respiratory tract, their function is going to go down. So you have increased secretions and decreased cilia activity due to this. And that's why pertussis is called whooping cough. All right? So to summarize, ADP ribosylation is a mechanism that some bacteria use, and it targets G proteins and disrupts their function. When it disrupts their function, this causes the pathology associated with these two toxins. And for the last slide, I want to talk about what happens when protein kinase A is successfully activated. So your signaling molecule bound to the G protein coupled receptor that will cause GTP, it'll act like a guanine exchange factor and cause GTP to bind the alpha subunit. The G protein is then going to come off of the receptor, and the alpha subunit is going to dissociate, and that's going to activate adenylyl cyclase. When adenylyl cyclase is activated, ATP is converted to cyclic AMP. The cyclic AMP can bind the regulatory regions of protein kinase A. And now here's the new part. Protein kinase A will travel to the nucleus, and it's going to phosphorylate CREB. So CREB is the cyclic AMP response element binding protein. So it's a CRE binding protein, okay? And when it's phosphorylated, it binds another protein called CREB binding protein. So that the end result is you have gene transcription. So the main part I want you to understand is that protein kinase A will phosphorylate CREB, and that phosphorylation of CREB allows it to be a transcription activator, okay? It's going to be a transcriptional activator that is cis-acting. All right, what does cis-acting means? It means that it's nearby or upstream of the gene that it regulates. All right, it's a transcriptional activator, and it is cis-acting. It is on the same side as this gene. All right. Okay, and that's all we need to know for now. Next time we're going to talk about GQ protein coupled receptors. We're going to talk a bit about calcium signaling and a bit about nitric oxide as well. Thank you.